Hello, my name is Madeline Walpley, and I'm going to be discussing meta-analysis of QTL for diverse traits in capsicum species chili peppers. Many of us are familiar with chilies for their delicious flavor and heat combinations, especially here in New Mexico, where our comfort food is spicy green enchiladas and spicy red tamales, but chilies also holds an economic and pharmaceutical value as well. When you have such an influential crop, it's important to maximize and control those beneficial traits for an optimal yield. Fortunately, with current genotyping technology, we are able to gather genetic information on these plants to better improve breeding. Quantitative trait loci, or QTL, which are genetic regions responsible for variation on quantitative traits, have become readily available in the past years. To that end, a QTL meta-analysis then combines all individual QTL mapping studies to further assess the genetic architecture of specific traits. Here we're going to be assessing QTL in capsicum species, and until now there have been no known meta-QTL studies of capsicum focusing on a variety of different traits. In this study, we performed a meta-QTL analysis to identify regions in the capsicum genome associated with important traits to be used for genetic improvement. Information identified from the study will be used to further develop DNA markers for improved breeding and selection of New Mexico chilies. The aim of this study was to develop a genetic consensus map for capsicum species, identify meta-QTL related with various traits, determine molecular markers linked to meta-QTL, as well as identify candidate genes present in meta-QTL regions. To obtain these goals, we first created a database containing 29 individual QTL mapping studies. These QTL were then projected onto a consensus map, which had been derived from 3 different genetic maps. From there, meta-analysis was performed using bioMercator, and meta-QTL were defined as those genomic regions in which at least 2 different QTL co-localized to form a meta-QTL. Finally, candidate gene analysis was performed using flanking marker sequences for meta-QTL with a less than 1 centimorgan confidence interval. We initially identified 766 individual QTL across 12 chromosomes. 90% of these reported QTL were identified as using either recombinant inbred lines or F2 biparental mapping populations. We focus on studies which use single nucleotide polymorphisms, amplified fragment length polymorphisms, and simple sequence repeats for marker types. Figure 1, as you can see, represents the distribution of traits which had been categorized into 5 different categories. We had adaptation, disease resistance, physiological and biochemical, agronomic and yield, as well as heat profile. As you can see, a majority of these traits were agronomic and yield, if you look at the orange bars there. But then if you look at a chromosome P5, for example, that contained majorly disease resistance related traits. Then we go down here and we actually performed the meta-QTL, in which we identified 30 meta-QTL. Chromosomes 1 and 5, they're stacked right on top of each other here, have the highest number of meta-QTL each with 4 meta-QTL regions. The position in centimorgans as well as the flanking markers were also identified. I know it is a little bit tricky to see there. A total of 346 candidate genes were identified using sequence information for the flanking markers for meta-QTL 5.1 and 5.2. These meta-QTL, again, were delimited to a confidence interval of less than 1 centimorgan, which is why they continued further for gene identification. From there, we determined biological functions for the candidate genes, which included those related with DNA replication, repair, transcription, regulation, phosphorylation, and glycosylation. Epigenetic mechanisms such as RNA, DNA, tRNA, and histone methylation have also been predicted for meta-QTL 5.1 and 5.2. In conclusion, we reported meta-QTL regions in chromosomes P5, which were related to P-capcc, fruit and root rot resistance, which correlated with potential epigenetic mechanisms which have actually already been recognized for their relationship with conferring disease resistance in other crops, for example, rice and maize, just to name a few. MetaQTL 5.1 and MetaQTL 5.2 could serve as potential targets for marker-assisted breeding for improved P. capsicum resistance in chili peppers. Meta-analysis identified 30 MetaQTL regions across the 12 chromosomes in capsicum. And from here, molecular markers based on these MetaQTL regions, i.e. 5.1 and 5.2, will be developed and validated for targeted marker selection using breeding lines from the NMSU chili pepper breeding program for improved P. capsicum resistance. Thank you.